Okay, hi everyone. I'm going to do a quick recording just to help you understand Monday's lab that we're going to be doing together, or it could be Tuesday if you're attend forensics on B block. So what I want you to do is, I'm going to zoom this out. I want you to pause the video and read through the case that you're going to be trying to solve on Monday. And again, these are kind of like our tests. You can use all of your notes. You can't do this if you don't have notes, so it's kind of a double whammy. If you didn't do yesterday's assignments and get the notes finished, then how can you help a group out in terms of answering and analyzing and performing this lab? So you just won't. You'll get a 0 on this, and you'll have a 0 on the notes. So make sure you have your notes done from the last class. Those notes were all about blood, also about blood typing and how we read the evidence so that we can determine a person's blood type, and it also has some information that you're going to need to answer the wrap-up questions. So again, pause the video, read through this so that you understand what this case is about and how to perform each step. Here is the analysis section. So this kind of tells you where our sources of blood are coming from. And then, again, according to these directions here, how you will record the results. Make sure you understand how you're going to record those. And then also, you need to obviously determine blood type. And that will help us maybe narrow down a suspect and the blood from the crime scene, etc. There will also be some other questions to answer, so you can look at those now, but you'll have a paper to fill those answers out on the end of the lab. Okay, so I'm going to quickly show you what the lab setup looks like, because then I won't have to explain it on Monday. And I won't explain it on Monday because you have this opportunity to look ahead. So this is using the document camera. You're going to get basically a blank piece of paper and then a plastic coated sheet. You'll put the paper inside the coated sheet. Now when we wash these coated sheets at the end of the hour, sometimes they lose their like a film that they have on them. So in that case, I'll give you a spray. So you will need to spray it when you start the lab, and then you will just rub that around with a paper towel. Again, you don't want to rub it off. You want to just rub it onto the plastic coating there. The next thing that you need to do is identify where your samples are coming from. So you'll use that lab sheet in order to do that. So remember, on this lab sheet, we can see that our first sample is coming from the crime scene. Then we have a victim sample of blood. Then we have our suspects. So you will use that. So what did I say the first one was? Sorry, blood sample. That popped up. So the blood sample, if the first one is coming from the scene, we can use a dry erase marker. Works well on these. We can just say that this is our scene sample, and then if we have our victim sample, and then we can go down the list, and then we can put our suspects. So then what we will do is we will pass around blood samples. So we've got these containers with blood samples in them. Victim, scene, depending on when you get it. If you get your victim sample first, then you'll start with that, and that's why we labeled everything. So we'll put one drop right onto where it says clarity test. One drop here for the B. And you got to be careful because that stuff we put on it kind of makes it bead up. So you want to, if it is slipping around, one of the cool things that you can do is you can kind of drag it back into its spot. And again, that's what that stuff that we sprayed on allows it to do. So if it is, you do not want them to merge together. So you do need to be careful with that. So I've got my samples of my victim blood. Once you have your whole chart filled out, and again, you got to be careful because if you do move it, they will sometimes slide around. So be careful. Once you've used a toothpick too, it is contaminated, so you need to make sure that you don't use that tip again. We can save a toothpick by just breaking off the tip, and then now we've got a clean end that we can use. You can get a lot of mixes out of a single toothpick. Okay? I just broke off the tip, so that way I'm not having to run and go grab lots of toothpicks. Plus I'm conserving and reducing my waste. The next thing that we would do, so I've got my victims A sample. Again, I'd have my scene. I'd have all my other suspect samples down. And then what we will do in class is we will start passing around antiserums. And so you should have read about this in the actual notes, but then also you are going to get a video, or you watched a video. I think I would have you do that first, the video on doing a real test of someone's blood. So I'll post both of those. But it kind of depends on what order you're watching the videos in. So, and then what we would do is we take the antiserum. So here I have antiserum A. It's called anti-A serum. And I'm going to place that. I'm going to do my 1 drop onto the blood for A. And since I'm going to actually have all of my blood filled out on this chart, I'm going to go down this whole row and fill that out. And then I've got my antiserum RH, it says on it, anti-RH. So that one would go over here. And again, 1 drop, being very careful because this stuff's expensive. And then I have my anti-B antiserum. So I would place that on here. And again, I want it to be on the words so I can see what's happening. I want to make sure that they don't merge into each other, so I'm being very careful. If I need to, I can kind of drag it back into here. Plus, I need to kind of mix these up. So that's fine. And again, don't reuse the toothpick, so break the toothpick. Mix this up here. You have to stir them or they might not react. So notice each tip is clean. Make sure they're on the words. And then I'm going to give this 10 minutes, 8 to 10 minutes to set. Honestly, the longer the better. By the time I get done with this, then it usually takes me a couple minutes to do all of my testing. So at that point, I can just set a timer for 8 minutes to make sure that everything's had enough time to fully react. And then I will, so like right now, I'm not necessarily going to see any observations because the full time has not passed. As time continues, there may be some agglutination reactions that are going to occur. Or this might be O negative and none of these will react. It really depends on the actual samples that I'm testing. So, and different groups will have different ones. So I couldn't even tell you what this one's supposed to do because you have different victims, etc. Well, I have different samples of victims. So anyway, so that's how we do these tests. And again, we give it a lot of time. And then we'll make our final analysis and fill out our data chart, and then we'll be able to answer our final questions in the big end of the lab.